जय हिंद गाइस, स्वागत है आपका हमारी फैमिली 24/7 सी जोन में। तो आज की हमारी बायोलॉजी क्लास का जो टॉपिक होगा वो है डीएनए रेप्लिकेशन, डीएनए प्रोसेसिंग एंड जीन रेगुलेशन। तो चलिए स्टार्ट करते हैं हमारी क्लास। क्वेश्चन नंबर 1 इज डीएनए सिंथेसिस टेक्स प्लेस ड्यूरिंग ऑप्शन ए इंटरफेज, ऑप्शन बी प्रोफेज, ऑप्शन सी मेटाफेज और ऑप्शन डी एनाफेज। So the right answer is option A, interphase. DNA synthesis takes place during interphase. Next question. Semi-conservative model of DNA replication was demonstrated by A. Herbert Taylor, B. Watson and Crick, C. Michelson and Stahl, or D. None. So the right answer is option C, Michelson and Stahl. So जो सेमी कंजर्वेटिव मॉडल ऑफ डीएनए रेप्लिकेशन था वो प्रपोज किया था वॉटसन एंड क्रिक ने, और जो डिमॉन्स्ट्रेट किया था वो था मिचेल्सन और स्टैहल ने। ओके, नेक्स्ट क्वेश्चन। Experimental material for the study of DNA replication has been A-pneumococcus B-Eschereria coli, C-Neurospora or D. None. So the right answer is option B, Escherichia coli. Next question. Isotopes used for proving semi-conservative replication of DNA is A. N14 and P31, B. N14 and C14, C. N14 and N15 or D. None. So the right answer is option C, N14 and N15. Next question. Taylor demonstrated semi-conservative chromosome replication in A. Vixia faba, B. Hala cell, C. Pisum sativum or D. None. So the right answer is option A, Vixia faba. Next question. DNA polymerase helps in A. Renaturation of DNA, B. Proofreading of RNA, C. Joining monomers of DNA or D. None. So the right answer is option C, joining monomers of DNA. Next question. In contrast to DNA polymerase, RNA polymerase edits as it synthesizes, works only in a 5' to 3' direction, synthesizes the RNA primer to initiate the DNA synthesis or D. None. So the right answer is option C, synthesizes the RNA primer to initiate DNA synthesis. Next question. DNA replication in eukaryotes commences A. from centromere to either end, B. several sites along DNA of chromosome simultaneously, C. both A and B or D. None. So the right answer is option number B, several sites along DNA of a chromosome simultaneously. Next question. The enzyme which cut the molecules of DNA into segments is known as A. DNA ligase, B. DNA gyrase, C. restriction endonuclease or D. None. So the right answer is option C, restriction endonuclease. और डीएनए लाइगेज क्या करता है? डीएनए के सेगमेंट्स को जॉइन करता है। तो जो जॉइन करेगा वो एंजाइम होगा डीएनए लाइगेज, और जो डीएनए को सेगमेंट्स में कट करेगा वो होगा रेस्ट्रिक्शन एंडोन्यूक्लियस. ओके, नेक्स्ट क्वेश्चन. ओकाज़ाकी फ्रेगमेंट्स appears in the course of A. repair of DNA, B. replication of DNA, C. hybridization of DNA, or D. all of these. So the right answer is option B, replication of DNA. The Okazaki fragments appears in the course of replication of DNA. Next question. The enzyme makes short RNA chains using a DNA template. A. Primase, B. DNA gyrase, C. DNA polymerase, or D. none. So the right answer is option A. Primase. The enzyme that makes short RNA chains using a DNA template is a primase. Next question. The enzyme which relaxes the supercoiled DNA is option A. primase, option B. DNA gyrase, option C. topoisomerase 1, or D. none. So the right answer is option C. topoisomerase. एज टोपोआइसोमेरेज क्या करता है, जो डीएनए के स्ट्रैंड्स होते हैं उनके बीच का टेंशन जो होगा उसको रिलीज करेगा. तो डीएनए स्ट्रैंड्स के बीच का जो टेंशन होगा वो रिलीज कौन करता है, टोपोआइसोमेरेज 1. दैट इज व्हाई द राइट आंसर विल बी ऑप्शन सी. नेक्स्ट क्वेश्चन. RNA is not found in option A. nucleus, option B. cell sap, option C. ribosomes, or option D. none. So the right answer is option B. cell sap. RNA is not found in option B. cell sap. Next question. A riboside is A. ribose plus base, B. ribose plus phosphate, C. base plus phosphate, or D. none. So the right answer is option A. ribose plus base. So ribose और base mix होके क्या बनाते हैं, riboside. Okay. Next question. DNA gyrase is inhibited by option A. tetracycline, option B. nalixidic acid, option C. both A and B, or option D. none. So here the right answer will be option B. nalixidic acid. The DNA gyrase is inhibited by nalixidic acid. Next question. mRNA is made of A. ribonucleotides, B. deoxyribonucleotides, C. both A and B, or D. none. So the right answer is option A. ribonucleotides. mRNA is made of ribonucleotides. Next question. Polymerase 3 catalyzes the synthesis of A. mRNA, B. tRNA, C. rRNA, or D. none. So the right answer is option B. tRNA. Polymerase 3 catalyzes the synthesis of tRNA यानी कि transfer RNA. Next question. Which site of a tRNA molecule hydrogen bonds to a mRNA molecule? Option A. codon, option B. anticodon, option C. 5' end of tRNA, or option D. 3' end of tRNA. So the right answer is option B. anticodon site of tRNA molecule binds to the mRNA molecule. Okay, एज एंटी कोडन साइट जो होती है वो टीआरएनए मॉलिक्यूल पे प्रेजेंट होती है, और जो कोडन वाली साइट होती है वो एमआरएनए मॉलिक्यूल पे प्रेजेंट होती है। ठीक है, नेक्स्ट क्वेश्चन। ट्रांसलेशन इज द प्रोसेस इन विच डीएनए इज फॉर्म्ड ऑन डीएनए टेम्पलेट, आरएनए इज फॉर्म्ड ऑन डीएनए टेम्पलेट, DNA is formed on RNA template, or protein is formed from RNA message. So the right answer will be option D, protein is formed from RNA message. As होता क्या है, पहले DNA से mRNA बनता है। इस प्रोसेस को हम ट्रांसक्रिप्शन बोलते हैं। ठीक, अब mRNA से जब प्रोटीन बनेगा तो इस प्रोसेस को हम ट्रांसलेशन बोलेंगे। ठीक, तो mRNA से जब प्रोटीन बना, यानी कि प्रोटीन का फॉर्मेशन जब होगा वो क्या होगा, ट्रांसलेशन। ओके, नेक्स्ट क्वेश्चन। विच इज नॉट इन्वॉल्व्ड इन प्रोटीन सिंथेसिस? ए, इनिशिएशन, बी, ट्रांसलेशन, सी, ट्रांसक्रिप्शन, और डी, नन। So the right answer is option A, initiation. Okay, next question. If one strand of DNA has the nucleotide sequence 5-GATCAA3', its complementary strand will have the sequence A 5-5-CTA or B 5-TTGATC3' or C 5-TTCACTC3' और D नन। सो इस क्वेश्चन को समझने के लिए सबसे पहले आपको जाना पड़ेगा डीएनए स्ट्रक्चर में। तो डीएनए स्ट्रक्चर में आपको पता ही होगा बेस पेयरिंग रूल क्या था कि A always pair with T and G always pair with C, यानी कि एडेनिन पेयर अप करता है थाइमिन के साथ और गुआनिन पेयर अप करता है साइटोसिन के साथ। ओके, सो इसका राइट आंसर क्या होगा, 5G है यानी कि गुआनिन है, तो पहले हम 5-N लिख लेते हैं। G है तो G किसके साथ पेयर अप करेगा, C के साथ। A है तो A किसके साथ पेयर अप करेगा, T के साथ। T है तो T किसके साथ पेयर अप करेगा, A के साथ। फिर C का हो जाएगा G, A का हो जाएगा T और अगेन T, देन 3'। So the right answer will be option A, 5-CTAGTT3'. Okay, next question. Adenine plus ribose plus phosphate forms A, adenosine, B, adenylate acid, C, adenosine diphosphate, or D, adenosine triphosphate. So the right answer is option D, adenosine triphosphate. Okay, adenine plus ribose plus phosphate forms adenosine triphosphate. Okay, next question. Pyrimidines of RNA is represented by A, uracil and guanine, B, adenine and cytosine, C, cytosine and uracil, or D, none. So the right answer will be option C, cytosine and uracil. Okay, next question. During transcription, the DNA site at which RNA polymerase binds is called A, receptor, B, promoter, C, both A and B, or D, none. So the right answer will be option B, promoter. During transcription, the DNA site at which RNA polymerase binds is called as promoter. Okay. Next question. Triplet codon refers to sequence of 3 bases on A, tRNA, B, mRNA, C, rRNA, or D, none. So the right answer will be option B, mRNA. एज मैंने अभी प्रीवियस क्वेश्चन में भी बताया था कि जो एंटी कोडन साइट होती है वो टीआरएनए पे होती है और जो कोडन साइट होती है वो होगी एमआरएनए पे. ओके. नेक्स्ट क्वेश्चन. द सक्सेसिव न्यूक्लियोटाइड्स ऑफ आरएनए आर कोवैलेंटली लिंक्ड थ्रू एंटी-पैरेलल ए ग्लाइकोसिडिक बॉन्ड्स, बी फॉस्फोडाइस्टर बॉन्ड्स, सी हाइड्रोजन बॉन्ड्स, और डी नन. So the right answer is option B, phosphodiester bond. The successive nucleotides of RNA are covalently linked through anti-parallel phosphodiester bonds. Next question. The prokaryotic DNA chain elongation is carried out by option A, DNA polymerase 1, option B, DNA polymerase 2, option C, DNA polymerase 3, or option D, none. So the right answer is option C, DNA polymerase 3. The prokaryotic DNA chain elongation is carried out by option C, DNA polymerase 3. Next question. Histones have an abundance of which one of the following amino acid? That is A, lysine and arginine, option B, alanine and glutamine, option C, glycine and glutamine, or option D, none. So the right answer is option A, lysine and arginine. Okay. Next question. The 5' and 3' number are related to the A, length of DNA, B, carbon number in sugar, C, both A and B, or D, none. So the right answer is option B, carbon number in sugar. The 5' and 3' number are related to the carbon number in sugar. Next question. The enzymes that is used for unraveling of short segments of DNA is option A, DNA polymerase, option B, helicase, option C, DNA ligase, or D, none. So the right answer is option B, helicase. The enzyme that is used for unraveling of short segments of DNA is helicase. Next question. Isolation and purification of specific DNA segment from a living organism was achieved by option A, Crick, option B, Nierenberg, option C, Khorana, or option D, none. So the right answer is option B, Nierenberg. Next question. Genetic information in DNA is coded in the A, number of bases, B, sequence of nucleotides, C, length of DNA, or D, number of nucleosides. So the right answer is option B, sequence of nucleotides. Genetic information in DNA is coded in the sequence of nucleotides. Next question. An aminoacyl synthetase is responsible for A. Formation of peptide bond, B. Joining of amino acid to a tRNA, C. Attaching an amino acid to group an organic acid, or option C. None. So the right answer is option B. Joining of amino acid to a tRNA. Next question. Inhibitor of protein synthesis that acts as an analog of aminoacyl tRNA is A. Rifampicin, B. Puromycin, C. Mitomycin, or D. None. So the right answer is option B. Puromycin. It is the inhibitor of protein synthesis that acts as an analog of aminoacyl tRNA is puromycin. Next question. The agent that triggers the transcription from an operon is a or an A. Inducer, B. Regulator, C. Depressor, or D. None. So the right answer will be option A. Inducer. The agent that triggers the transcription from an operon is a or an inducer. Next question. Operon model for regulation of transcription was proposed by A. Jacob and Monod in 1961, B. Michelson and Stahl in 1998, or C. None. So the right answer is option A. Jacob and Monod in 1961. Next question. Balbiani rings are the sites of A. RNA and protein synthesis, B. DNA replication, C. Synthesis of lipids, or D. None. So the right answer is option A. RNA and protein synthesis. Balbiani rings are the sites of RNA and protein synthesis. Next question. The enzyme reverse transcription is A. RNA dependent RNA polymerase, B. RNA dependent DNA polymerase, C. DNA dependent DNA polymerase, or D. None. So the right answer is RNA dependent DNA polymerase. That is option B is the right answer. The enzyme reverse transcription is RNA dependent DNA polymerase. Next question. Reverse transcription was discovered by A. Kornberg and Mattei, B. Temin and Baltimore, C. FHC Crick, or D. Nierenberg, Khurana and Holle. So the right answer is option B. Temin and Baltimore has discovered reverse transcription. Okay. Next question. Chain initiation or starting codon is option A. UGA, option B. GUG, option C. AUG, or option D. None. So the right answer will be option C. AUG. Next question. In operon model, regulator gene A stops the transcription, B inactivates the substrate, C stops the formation of RNA, or D. None. So the right answer will be option A. Stops the transcription. Regulator genes का फंक्शन क्या होता है? ट्रांसक्रिप्शन को स्टॉप करना. ठीक. Next question. How many E. coli is in the lactose medium? Option A 3, option B 5, option C 6, or option D none. So the right answer will be option B 5. Next question. Which of these is associated with nucleolus? A. tRNA, B. rRNA, C. both A and B, or D. none? So the right answer is option B, rRNA or ribosomal RNA. Next question. Which RNA is related with clover leaf model? Option A, tRNA, option B, mRNA, option C, both, or option D, none. So the right answer will be option A, tRNA. tRNA is related with clover leaf model. Next question. In operon model, RNA polymerase binds to option A, structural gene, option B, regulator gene, option C, operator gene, or option D, promoter gene. So the right answer is option D, promoter gene. In operon model, RNA polymerase binds to promoter gene. Next question. The repressor is able to bind to the operator in the presence of which amino acid? A. Tryptophan, B. Histogen, C. Lysine, or D. none? So the right answer is option A, tryptophan. The repressor that is able to bind to the operator in the presence of the amino acid tryptophan. Next question. In eukaryotes transcription, the RNA splicing and RNA capping takes place in A. ribosomes, B. nuclease, C-dictyosomes, or D. none? So the right answer is option B, nucleus. In eukaryote transcription, the RNA splicing and RNA capping takes place in nucleus. Next question. Two strands of DNA are A. similar in nature, B. antiparallel and complementary, C. dissimilar and non-complementary, or D. none? So the right answer is option B, antiparallel and complementary. Two strands of DNA are antiparallel and complementary. Next question. Which antibiotic inhibits the interaction between tRNA and mRNA during bacterial protein synthesis? So option A will be streptomycin, option B tetracycline, option C erythropoietin, or option D neomycin. So the right answer is option D, neomycin. Neomycin is the antibiotic that inhibits the interaction between tRNA and mRNA. Okay, lac operon is related with A. synthesis of lac by lac insect, B. synthesis of enzyme of lactose catabolism, C. both A and B, or D. none? So the right answer is option B, synthesis of enzyme of lactose catabolism. Lac operon is related with synthesis of enzyme of lactose catabolism. Okay. सो आज के लिए बस इतना ही. वीडियो अच्छी लगी हो तो लाइक करना, फैमिली में नए हो तो सब्सक्राइब करके ज्वाइन करना. आज की वीडियो में बस इतना ही. मिलते हैं नेक्स्ट क्लास में किसी न्यू टॉपिक के साथ. तब तक के लिए पढ़ते रहिए और जो भी पढ़ा है उसको रिवाइज ज़रूर कीजिए. ओके. जय हिंद, जय भारत.